to you about some of the important questions that researchers have in studying TLRs today. How to tease apart complex TLR pathways, and a key consideration, what signaling mechanisms are you trying to study? Novus has a wealth of research tools for analyzing TLR expression as well as inhibiting and activating the TLR pathways. Some of the tools that I'm going to talk about today include TLR antibodies as well as TLR pathway antibodies, ligands for activating TLRs, inhibitors for inhibiting the pathway, stable reporter cell lines to study the mechanisms of TLR activation and inhibitors, as well as clinical tissue lysates from patient donors that have different types of cancers. So as most of you probably all know, TLR receptors were first defined as key regulators of innate immunity. However, they were later found to also have broad roles. So that we are all on the same page, I want to start with a TLR signaling pathway overview, followed by TLR activation and inhibition assays, and then finally I will talk about the emerging awareness of TLR post-translational modifications and how that can affect Western blot analysis. Here's an overview of TLR signaling pathways from our latest TLR signaling pathway poster, and if you don't already have a copy of this, I would encourage you to request your complimentary copy. As you can see from this poster, TLRs may be at the surface of membranes or in endosomal membrane components of the cell. In any case, TLRs are activated by ligands, which result in TLR complexes that are formed with TLR cofactors and adaptor proteins. This leads to activation of downstream signaling through the NF-kappa B pathway or other pathways and ends up with pro-inflammatory gene expression. I would like to point out a few things about this poster. Number one is that you can see these various stop signs, and later I'm going to talk to you about inhibitors that we have that will specifically stop TLR signaling at these nodes. Additionally, I would like to point out that all of the TLRs signal through adaptor proteins, and you can see that here that there's different proteins including TRAP and TRAM, as well as a common adapter called MyD88 where the TLRs, except for TLR3, all signal through. So let's begin on our journey about TLRs and TLR activation. So from the poster, I showed you that TLR activation leads to immune responses. These immune responses play key roles in innate immunity. They also bridge to adaptive immunity, and it is increasingly being recognized that TLR dysregulation has key roles in autoimmunity and cancer. And I want to start and talk about that by an example of TLR activation and the expression of psoriasin, which is involved in both innate immune responses as well as autoimmunity and cancer. So here's a cartoon that shows the activation of TLR5 by flagellin. Flagellin is a component of bacterial proteins, and so it's a TLR5 ligand, and when it binds to TLR5, TLR5 is activated, and among other things, psoriasin can be upregulated. Now, the upregulation of psoriasin is important because one of its properties is microbicidal actions. And so if you are exposed to pathogenic bacteria, you might like to upregulate microbicidal proteins such as psoriasin on the surface of your skin, for example, to kill these microbes. However, in addition to normal innate immune responses, the upregulation of psoriasin is found with various immune disorders. The most commonly recognized is psoriasis. So in psoriasis, when people have psoriasis outbreaks and scaly skin patches, these patches have an abundance of psoriasin in them. And so this is thought to contribute to inflammation and the pathology of the disease. Likewise, most of you probably know that it's increasingly being recognized that there are links between inflammation and cancer, and psoriasin is one of these proteins that is thought to be involved in cancer. And so I want to talk about a tumor model called breast ductal carcinoma, which is used to study the upregulation of psoriasin in breast cancer, as well as some other cancers that it is now being discovered in. So we have tools called clinical tissue lysates, and these are from actual patient donors, whereby in the course of their treatment, specimens are taken which include the tumor as well as the normal adjacent tissue, which is defined as tissue outside of the margins of the tumor by a pathologist. Now, the normal adjacent tissue as well as the tumor is processed and manufactured into lysates, being careful to avoid this necrotic area that is common in many tumors. And so these lysates are available as native lysates as well as ready-to-use blots with tumor and normal adjacent lysates in alternating rows of 7 patients. So here's 7 different donors where there's samples of the tumor as well as the normal adjacent tissue. And so the first example I want to show you is using these blots to probe psoriasin. Psoriasin is, as we talked about, a protein that is upregulated during TLR5 activation and also happens to be found in high levels in ductal carcinoma. In fact, this protein was independently discovered in ductal carcinomas as HID5, which stands for high in ductal carcinoma, and later was found to be the same as psoriasin, which was first discovered in patients with psoriasis. Now, in probing 7 different pairs of patients, tumor, normal adjacent tissues, we can see a clear pattern emerging here in that we see a detectable level of psoriasin in many of the tumors. However, when we look at the normal adjacent, we see a lack of psoriasin expression. So by using 7 different patient donors, you can see that there's a variable expression in these patient donors. We're used to all studying cell lines where, for example, HeLa cells, where we have certain expectations that cancers may have high levels of expression. But when we go to actual patients, we can see that there's quite a bit of variability. So we're very excited about these research tools, which will enable us to probe different types of donors. And if a researcher, for example, is using these tools, they may, for example, be interested in this patient, number 5, which has detectable levels of psoriasin in both the tumor and normal adjacent tissue, and might want to look at this lysate and probe to see what other biomarkers go with it. Likewise, they may want to look at another patient, say, for example, number 2, and say, well, what changes have happened along with psoriasin upregulation from the normal adjacent to the tumor tissue? Or they might want to look at another pair which has rarely any or hardly any detection of psoriasin in either the tumor or normal adjacent tissue. Likewise, we can use these tissues to also probe for TLR expression, and I want to make a point here that there's variable expression of different TLRs. For example, in these colon tissues where there is 5 different colon donors as well as a donor with ovarian cancer, we can see the expression of TLR3 in both the tumor and normal adjacent tissue of every patient donor. However, when we look closely, we can see qualitative, if not quantitative, differences in some of these patients. For example, in the normal tissue of patient 2, we see a broad band of TLR3 expression, which may represent different patterns of glycosylation in comparison to the tumor of this patient, where you have more of a single band. And so, likewise, in the ovarian normal tissue, we can see that the band appears to be broader than in the tumor adjacent tissue. So we can start to see that there's different patterns of TLR expression in the different patient donors, and what that means would be to follow up and perhaps look at other biomarkers to ask what is the difference between the normal adjacent and the tumor tissue of these different patients. In contrast, when we look at TLR1, we can see quite a variability in TLR1 expression in that the patients, some of them have no detectable expression of TLR1 in either the tumor nor the normal adjacent in patient number 3. And then in patient number 1, we can see full-length TLR1 expressed only in the tumor, but not in the normal adjacent. And I'd like to point out here the value of actually looking at the entire Western blot, because if we just looked at the expected molecular weight, we may miss some of these cleavage fragments, which could be important, and we will take this up in the third part of the presentation. In addition to probing Western blots, it's interesting to look at TLRs in the local microenvironment by immunohistochemistry. So here's the TLR4 probed in 4 different patients, and this is actually from a publication from 2013 by Zhang et al., looking at 4 different stages of TLR4 in 4 different patients. And you can see quite a difference in the variability of expression of TLR4, both in the amount and in the cell types expressed. And in his publication, he talks about how TLR4 can be expressed not only in the tumor cells, but also in the microenvironment. And in fact, we are increasingly recognizing that there is important interplay between the tumor and the microenvironment. It used to be thought that we would just look at dysregulation of a given protein in the tumor, for example, but now we know that some of the infiltrating cells, such as the lymphocytes or other immune cells, may have aberrant or high levels expression of TLR, and likewise, there can be changes in the normal adjacent tissue. For example, I showed you in the Western blots how some of the normal adjacent tissues of some patients had expression of the TLRs or psoriasin, but other patients did not. And so these tools actually enable you to probe at a deeper level and see what other changes may be important. And I just want to leave this section with a quote from Angela Coleman from the 2014 Advancement of Science meeting in Chicago, where she talks about that we're increasingly recognizing that we can harness the immune system to manage cancer. However, we still need a more sophisticated knowledge to really work this through. And we're hoping that Novus actually will provide researchers with top-level tools to look further into this. So we talked a little bit about innate immunity with the psoriasin response and disease, and I want to move here towards a cartoon which shows that it used to be thought that there was a clear distinction between innate and adaptive immunity. People used to think, well, there were cells of the innate immune system and there were cells of the adaptive immune system, and that TLRs were belonging to innate immunity. However, we now know that innate immunity and adaptive immunity are not so separate, and that the TLRs can actually bridge innate and immune adaptive responses. And I just show you one example here whereby in the classical innate immune system we have TLR activation through the antigen presenting cells, and the antigen presenting cells then bind to the T cells, which then cause downstream inflammatory responses. And then these downstream inflammatory responses then bridge back and affect the innate immune system. So this is sort of a modern day view of innate versus adaptive immunity, and kind of rethinking the dogma that these two immune systems are actually very intertwined. And TLRs play a key role in this entire process. So just to summarize before we move on to activation and inhibition assays, I showed you an overview beginning with the TLR signaling poster, which showed you how signals through TLRs activate downstream pathways. These lead to normal immune responses, and also dysregulation may lead to disease roles if you have overactivation of TLRs and overexpression of proteins or constitutive expression of those downstream proteins without TLR activation. Now I would like to move to activation and inhibition assays. And I show you this cartoon which really mimics the poster, where we show that ligands result in TLR activation. TLR activation results from a complex called the activating signaling complex, which forms upon ligand activation, and is this signaling complex that then leads to downstream signaling. So if this adaptor TLR complex is disrupted by inhibitors, these signals cannot proceed. This is essential for signals traveling through TLRs. So how can we look at this and start to tease apart TLR signaling complexes? And people use this type of model system to ask the question, which TLRs may be involved in my system, which pathway specifically may be involved, and another application may be through drug screening, applying drugs through cell lines to see which TLR pathways might be activated. So here's an example showing the TLR4 pathway and using a TLR4 reporter cell line to measure responses. So here you can see that TLR4 is activated by LPS, leading through downstream responses, and instead of cytokine secretion, we have cloned in the stable cell line the NF-kappa B response element. So when you have all this activation, it activates this response element, which transcribes CEAP, and CEAP is secreted into the media, and that is actually what is measured. So in this model system, which I'm going to show you an experiment of, we used LPS as the ligand to activate TLR4. We used Viper as an inhibitor, which binds and disrupts the tier-tier domain interaction between TLR4 and its adaptor proteins TRAM and TRAP to stop propagation of the signals and thereby result in a lack of CEAP production. So how does this exactly work? Well, all the inhibitory peptides that I'm going to talk to you about today are based on sequences which contain an inhibitory part as well as a cell permeable part. And the cell permeable part allows the sequence to passively get into the cell when incubated into the media. And then the sequence, the inhibition sequence, in this example, it binds right here and disrupts this tier-tier domain interaction. And other inhibitors, which I'll show you, have other specific inhibitory features. And then we have in each inhibitory peptide the cell permeable domain, and in this case, the cell permeable domain is a scrambled sequence, and so it is not expected to bind here, but rather it just gets into the cell and serves as a control. Now, Viper stands for Viper inhibitory peptide of TLR4, and it's from the A46 vaccinia sequence, and this virus actually, this is how it disrupts normal cell machinery by actually binding here and disrupting your TLR4 functions, and so you cannot mount the normal inhibitory response against the vaccinia virus, and that's how you become sick when the viruses attack your body and you can't have the proper immune response. So we can conveniently use this TLR reporter cell line to study this. And I want to focus on this graph here where we have the inhibitory peptide in red and the control peptide, which we call CP7, in blue. And so you can see that in the absence of LPS activation, we just have basal levels of CEAP production. And why are there basal levels? Well, CEAP is attached to NF-kappa B, and when you have cells in culture, you can have a basal level of activation of NF-kappa B just through normal cell functioning. However, when you add LPS, you can see that the CEAP signal dramatically increases. And so both the Viper and CP7 were not added here. They were at 0 concentration. So this is actually the maximal response that we've determined for LPS activation through other studies. And then when we add increasing amounts of Viper peptide in red, you can see an inhibition of the LPS response. Now, it's important to incubate Viper into the cell prior to adding LPS because it does take some time for Viper or the control peptide to cross the membrane and get into the cell. And in this case, when Viper gets into the cell, it will bind to those tier-tier domains. And so when we activate with LPS, we will have an attenuated response. Likewise, you can see using the control peptide at the same concentration did not affect the response. And down here, it just shows the IC50, the concentration where the inhibitor response is reduced by half. And that's because even when you add more and more inhibitor, you've saturated everything. And so you want to make sure not to add too much and to determine the optimal concentration for your model system. So that's an example of TLR4 inhibition, I want to move now to MyD88. Now, most of you probably know that MyD88, which is shown by stop sign 1 here, the inhibitor that inhibits MyD88 homodimerization, is an inhibitor used by most all of the TLRs except for TLR3. So this is a more downstream inhibitor than, say, inhibiting TLR4 right at the TLR4 upstream adapter of TRAM or TRAP, and also it will inhibit more than TLR4 signals. So what I've used as a model system here is TLR5. And before I go to showing you inhibition, when you're doing your studies, you must make sure that your ligand is actually activating your cells. And so here we show Flagellin added to the TLR5 cell line, and you can see that Flagellin as a ligand for TLR5 activates TLR5 as indicated by SEEP production. And once again, we can determine here the EC50, which is the ligand concentration that gives half maximal response. So this is important to determine because if you add too much ligand to your system and you overwhelm the system and you add a little bit of inhibitor, it won't be able to stop all the responses. So we always encourage researchers to do these titrations to determine what their EC50 is and then base their ligand concentration on that. So let's look at the experiment by using the MyD88 inhibitor. So here you can see the Flagellin activating TLR5 and the MyD88 inhibitor, which prevents this homodimerization and the downstream pathways. And so when you add this inhibitor and you look at the inhibition response, you can see that increasing amounts of peptide reduce the SEEP response compared to the control. So this is what I mean as how you must find the optimal inhibition, because if you add too much, you start to get nonspecific effects, or you may get nonspecific effects of the control because it is in itself a peptide. And then the inhibitory peptide then also can have maximum responses because once you've saturated the sites, adding more isn't going to help you any. So this cartoon just summarizes the MyD88 inhibitory response whereby we prevent this homodimerization, and so the responses can't continue. And this peptide is actually our most highly published peptide. And in looking through the literature, we can see that people used various readout systems to measure its inhibitory effect. I showed you the SEEP readout system. However, I've also seen it used with a CAT reporter. I've also seen people look at the inhibition of phosphorylation levels because we know in the TLR pathway there's many phospho effects, and so if it's inhibiting downstream effects, it may affect phosphorylation at a certain level. People look at mRNA expression, Western blot. Perhaps the most common is looking at cytokine ELISAs or looking at the inhibition of cytokine production because we know that TLRs lead to cytokine production. So it is up to each researcher to determine what readout system they want to look at. We offer the TLR reporter cell lines as controls for looking at these inhibition responses. So I showed you upstream inhibition responses closer to the TLR pathways. Now we can also look at inhibition downstream. We know that NF-kappa B is a key component of the pathway activation in TLR functioning, and so the signals that propagate through NF-kappa B activation can be stopped at different points of the pathway. And here's a little bit bigger view where we can see we can stop IKK formation, we can stop signal, the NSL signal localization unmasking, or we can stop phosphorylation of P65. And so let's focus in here because this is the example that I want to show you, stopping phosphorylation at serine 276 or 529. And so how do these peptides work? Well, these peptides contain either serine 276 consensus sequence or consensus sequence for serine 529 and 536. And so how do these work? Well, when you add one of these inhibitory peptides into your system, the peptide will be phosphorylated at this consensus sequence, and therefore your endogenous P65 protein won't be phosphorylated, and so that will stop a phosphorylation which may be important for NF-kappa B activation in your system. And so I showed you before with the other examples about how we inhibited responses, but you may have noticed that we didn't inhibit 100% of the responses, and people might ask about that, and they often do. So why is that? Well, all of these signaling pathways are very, very complex, and I thought I'd show you NF-kappa B as a good example, and this is from an excellent publication, which I would encourage you to look at. Here are all the post-translational modifications that may occur in P65 on its way to activation, including various serine phosphorylations and different acetylations. So you can imagine if you are just stopping one of these, it may not stop the entire response, or depending on your model system, it may not stop the response at all because your model system may not utilize that particular phosphorylation site. So it's very important to research and know something about the model system which is being used, and also when interpreting your data and wondering why the response is more or less depending on your model system. So suffice it to say, in this model system of TLR5, we can see that when we add the serine 276 inhibitory peptide activated by Flagellin, that we have an inhibitory response which is about 25 to 40% with this serine 276 peptide. So we can conclude that in this model system that serine 276 is important in the activation of TLR5 by Flagellin. However, there are also other mechanisms in action, which means that this does not totally stop all NF-kappa B activation. So here's a summary just showing you the different inhibitors that we have, and I would just like to point out that today we talked about MyD88 homodimerization inhibition. We also talked about tier domain inhibition, and we talked about inhibition of phosphorylation. We also have peptides that inhibit the IKK complex as well as inhibit the nuclear localization unmasking of P50. And we have, in addition to Viper, we have tier domain inhibitors which inhibit both TLR4 and TLR2. The Viper is specific for TLR4. So far, we've talked about TLR signaling overview, how TLR signaling happens. We talked about how we can activate and inhibit the TLR pathway with ligands and specific inhibitors in combination, and we can use reporter cell lines to look at these responses, as well as I talked to you a little bit about the published literature, which talks about various readout systems, and actually every single one of our inhibitors is in the published literature, and I would encourage you to look more about that. Now, I would like to talk to you about emerging awareness of TLR post-translational modification and how that affects Western blot analysis. This is a very, very common topic where people call in and say, well, I wanted to see a single band at a certain molecular weight and I didn't see that, and what do I do? And when we look at Western blots, and I'm sure most of you who do Western blots know this, that you see, if you look at the entire blot, you can see many, many bands often. And however, what people do is they publish, they cut out a little section of their blot and they publish like this, and they show only one band, and that really perpetuates the myth that a given protein is identified as a single band at a particular molecular weight on a Western blot. However, we can see here, and this has built-in controls really where we see it not binding in some cancers, some colon cancers, binding in other colon cancers, and we can see the pattern. In this individual, there was only a single band. In this individual, there was multiple bands. Here, there was only cleavage. And so if we cut out bands, we may miss some of this important data, and we may not yet know what the importance of this data means if we only show bands like this. So in looking through the literature, we can actually find that the TLRs are published with a number of important findings, and I'm just showing you a few examples. For TLR4, for example, we found a paper where they looked at urinary outputs, and they found that there were two different TLR9 bands reflecting different degrees of TLR4 glycosylation. So the more glycosylated, the heavier band. It may view as a broad band or else two different bands. And importantly, in post-ischemic urine samples, they saw only a 30 kilodalton band, and this reflected a presumptive TLR cleavage product. Now, people not reading this paper might think, oh, I only see a 30 kilodalton band, so I'm not seeing any TLR4. But when we go to look at the literature in this very careful study, these researchers found that this was actually a cleavage fragment of TLR4. Likewise, when they treated with cisplatin, they noticed that after 18 hours, they saw both 30 kilodalton and 60 kilodalton forms. However, after 48 hours, only the 30 kilodalton form was present. So just as an example of the importance of looking at your entire blot and researching the literature to see what the different banding patterns might mean. Likewise, it's increasingly being recognized that cleavage is important in the functioning of TLRs. In fact, for TLR9, it is thought that the cleave band is actually the functional band. And in this study, the researchers observed both full-length 120 kilodalton and 72 kilodalton cleave bands. And the underlying biology of the cleavage and different post-translational modifications is just beginning to be elucidated. For example, in this study in Huvette cells, the TLR3 was detected as 130 kilodaltons, which we usually think of full-length TLR4. However, when they treated with interferon beta, they got a higher molecular weight band, which was found to be due to glycosylation higher than 130 kilodaltons. Likewise, when cells were transfected, so they know they have TLR3 in there for sure, and they saw it at 130 kilodaltons, and when they expressed a cogene with it, UNC93B1, they got a higher molecular weight band of TLR3 due to glycosylation. Likewise, when they treated with this peptide glycosidase, the TLR3 band was less than 100 kilodaltons. So I think it's important to think about the banding patterns and what that may mean when doing Western blots, rather than, for example, jumping to conclusions and thinking that you may have seen the wrong size because you may miss important information. And I just want to point out with this that the size that you see may depend on the cell type or model system that you're using. For example, in the Huvette cells, I showed you that TLR3 was detected at 130 kilodaltons. But in this paper, TLR3 was detected at 108 kilodaltons in granulosa cells. And so the researchers wondered about that, but they validated the antibody in Western blot in their knockout and wild type animals, where they found that this 108 kilodalton band disappeared in their TLR3 mouse model system. So we're really just beginning to have awareness of TLR3 and TLR4, TLR9 post-translational modifications, as well as other TLRs. And I've just kind of compiled some information from our own observations, from those of our customers who write and ask us about banding patterns, as well as the literature. TLR banding patterns may vary in Western blots. You may see cleavage, glycosylation, varying molecular weights. Most all of our TLRs are highly validated and highly published. And I would encourage you to look at the Western blot publications that we have on our data sheets, because we've taken a lot of time to actually detail some of that. And likewise, every month more and more publications are coming out. And so if you don't see what you're looking for, going to scholar.google.com, you can search by catalog number, clone name, or you can write into us at technical service and we will help you do that. So this is a story told for today. I hope you enjoyed the TLR signaling overview and how you can use our research tools for detecting TLRs, how you can use our clinical tissue lysates for investigating TLR expression in different patient donors, how we can use our ligands and inhibitors to activate and inhibit the pathway, as well as giving you some insight into the emerging awareness of TLR post-translational modifications, which is definitely important if you plan to do Western blots. And The tools that I've discussed, the antibodies, the ligands, the inhibitors, reporter cell lines, and lysates are all available on our website. And if you need additional help finding them, please send us an email, contact technical or customer service, and we will be happy to help you.